Assalamu alaikum. Today our lecture will be about nerve tissue, the last lecture in this semester. The objectives are to describe the structure of the nerve tissue, to identify the types of neurons, to describe the structure and types of synapses, to identify the types of glial cells. The contents of this lecture are neurons, classification of neurons, synaptic communication, glial cells of the peripheral nervous system, glial cells of the central nervous system, and neuropil. The two principal cell types of the nervous system are neurons, which are excitable cells that transmit the electrical signals. The second is glial cells, which are supporting cells that surround and wrap neurons. We will discuss each of these types of cells in details in the following slides. First, we will start with neurons. The functional unit in both the central and peripheral nervous systems is a neuron. And by the central nervous system we mean the brain and the spinal cord. The peripheral nervous system we mean the cranial nerves, spinal nerves, and the peripheral nerves. So the functional unit in both systems, the peripheral and central nervous system, is the neuron. And the neurons consist of three main parts. Each neuron has three main parts. The first is the cell body, which is called perikaryon, okay, or soma. The cell body, perikaryon, and the second part, the dendrites. The third part is the axon, and we will talk about each one separately. In this image, we'll see this how the shape of the neuron appears in this diagram. It has a cell body, axon, and dendrites, the dendrites which emerge from the cell body. And here in the right side we have histological section of a neuron. Also we have these dendrites, cell body or perikaryon, and axon. What is the perikaryon or neuronal cell body? It contains the nucleus and the nucleolus. It is a part of the neuron that contains the nucleus and the nucleolus and the organelles. And it has a well-developed rough endoplasmic reticulum, which appears in histological sections as basophilic substance, chromatophilic substance, called Nissl substance or Nissl bodies. They appear as basophilic clumps in the histological sections. They are composed of concentrated rough endoplasmic reticulum. Also the perikaryon, it contains axon hillock, which is a cone-shaped area from which the axons arise. الحتة اللي بيطلع منها الأكسون is called the axon hillock, and it's part of the perikaryon or the cell body. The perikaryon or the cell body, it serves or functions as the synthetic or trophic center for the entire neuron. As we see in the left this diagram, this is the neuron perikaryon or cell body. It's a biosynthetic center and receptive region, okay, and it contains the nucleus and the nucleolus, and we have the Nissl bodies here. They are rough endoplasmic reticulum. On the right side we have a histological section, and here we will see the perikaryon, the nucleus, and the NS stands for Nissl bodies or Nissl substance around the nucleus. And here we have an axon hillock. The axon hillock is the region where the axon arise. The axon hillock, AH stands for axon hillock, and the these are the dendrites. Okay, in the diagram we'll see the axon arise from the axon hillock here, part of the cell body. What is the dendrites, or let's talk briefly about dendrites. They are short, tapering processes extending from the perikaryon, and they are the receptive or input regions of the neuron. Dendrites, they are the receptive region or the input region of the neuron. They receive the impulses and they act as receptors. They receive the stimuli from other neurons at unique sites called synapses. So they receive the stimuli or the impulses from other neurons at unique sites. They receive the stimuli from other neurons. What's axon? Or it is a single long process of uniform diameter arising from the hillock, from the axon hillock at the neuronal cell body. It generates and transmits action potentials. The axon generates and transmits action potentials, and it secretes neurotransmitters from the axonal terminals. Each axon has an axonal terminals, and it secretes neurotransmitters. Now let's talk about the classification of the neurons, the structural classification. We have anaxonic neurons, unipolar or pseudo-unipolar neurons, bipolar neurons, and multipolar neurons. The anaxonic neurons, they don't have a definite axon. There is a multiple dendrites. The unipolar or the pseudo-unipolar neurons, there is only one process that branches into two branches. One branch directed towards the central nervous system, the short one, and the longer one directed towards the peripheral sides of the body. The bipolar neurons, we have two processes, one dendrite and one axon. The bipolar neurons, and the multipolar neurons, they have multiple dendrites and in addition to the axon. In this diagram we will see the types of the neurons. The first one is the anaxonic neuron. We have many dendrites, but there is no true axon. There is no axon. In the bipolar neuron, we have one dendrite and one axon arising from the cell body. In the unipolar neuron or pseudo-unipolar neuron, we have single process that branches into two. The longer branch extending towards a peripheral ending, and the other branch heading towards the central nervous system. So in the pseudo-unipolar, we have a single process arising from the cell body, and then it branches into two. One branch towards the central nervous system and the other branch towards peripheral ending. And the multipolar neuron, this is how it looks like. We have one axon with multiple dendrites. Okay, multipolar neuron, we have one axon and multiple dendrites arising from the cell body. The multipolar neurons, they are the most, this is the most common shape of a neuron, or the most common type of neuron, the multipolar. Most neurons are multipolar. The bipolar neurons are found in the retina, the olfactory mucosa, and the cochlear and vestibular ganglia. That means bipolar neurons are concerned with Retina, that means vision. Olfactory mucosa is smell, concerned with vision, smell, and hearing, and balance. Cochlear, the inner ear, and the vestibular ganglia, or the balance. Pseudounipolar neurons are found in the spinal ganglia, in the peripheral nervous system, in the spinal ganglia, and in most cranial ganglia. Regarding the functional classification of the neurons, we can classify the neurons into sensory afferent neurons, which transmit the impulses toward the central nervous system. They carry the impulses toward the central nervous system, toward the brain and spinal cord. And the motor efferent neurons, they carry the impulses toward the body surface. They carry the impulses from the central nervous system towards body surface. The third type is the interneurons or association neurons. They are any neurons between a sensory and a motor neuron. We have finished the classification of neurons. Now we are going to talk about the synaptic communication, how the neurons they communicate. The synapses are the sites where nerve impulses are transmitted from one neuron to another or from neurons to other effector cell. And what are the components of a synapse? Each synapse has a presynaptic axon terminal or terminal button, and we have postsynaptic cell membrane, and the third part is the synaptic cleft. In this diagram, we will see how the components of the synapse appear. This is the axon of presynaptic neuron with the axon terminal or terminal button, and here we have the synaptic cleft, this potential space, and we have here the postsynaptic neuron. The presynaptic axon terminal contains synaptic vesicles, many vesicles containing the chemical neurotransmitter. In this diagram, it's acetylcholine. We have these vesicles containing the chemical neurotransmitter. We have calcium ion channels, they are voltage gated. And on the postsynaptic neuron, we have also we have the receptors for acetylcholine. Okay. So the postsynaptic cell membrane contains the receptors for the neurotransmitter released at the synaptic cleft from the axon terminal, from the presynaptic axon terminal. The types of synapses, we have axosomatic, axodendritic, and axoaxonic. In this diagram, we will see the difference. The axosomatic synapse, it's a synapse between axon and a cell body. Axon of presynaptic neuron and a cell body of postsynaptic neuron. This is called axosomatic synapse. The second type is the axodendritic synapse, which is a synapse between an axon of presynaptic neuron and the dendritic spines or the dendrite of postsynaptic neuron. So this is axodendritic synapse. The third part is axoaxonic synapse. It's a synapse between axon of presynaptic neuron and axon of postsynaptic neuron here. This synapse is called axoaxonic, between axon of presynaptic neuron and axon of postsynaptic neuron. Here is the synaptic cleft. So we have three types: axosomatic, axodendritic, axoaxonic, according to the communication and the synapse between which parts of the neuron. Axon and cell body, axosomatic, axon and dendrite, axodendritic, axoaxonic, axon and axon, it's called axoaxonic. Now we have finished the synaptic communication and the types of synapses and the components. We are moving to talk about the glial cells, or the supporting cells, called neuroglia. Yeah, the glial cells, they provide a supportive scaffolding for neurons, and they segregate and insulate neurons. So they are supportive for the neurons, and they provide an insulation for neurons. We have glial cells for the peripheral nervous system, and they are different from the glial cells of the central nervous system. In the peripheral nervous system, we have Schwann cells, or called neurolemmocytes. They produce myelin and insulate the nerve fibers. The other type is satellite cells of the ganglia, and they surround the neuronal cell bodies located within the ganglia and regulate the environment around the neurons. They are found around the cell bodies, neuronal cell bodies within the spinal ganglia, peripheral ganglia, and they regulate the environment for these neurons. As you see in this diagram, we have a sensory neuron with Schwann cells and satellite cells. These are the Schwann cells. They form myelin sheath that covering the axon, and we have here around the cell body of the neuron satellite cells. These are found around the neuronal cell bodies in the ganglia, and we have in the ganglia pseudo-unipolar neurons. The myelin sheath is a whitish fatty lipoprotein segmented sheath around most long axons, and it functions to protect the axon, to electrically insulate the fibers from one another, and increase the speed of nerve impulse transmission. So the myelin sheath protects the axon, electrically insulate the fibers, and increase the speed of nerve impulse transmission. This sheath, myelin sheath, is segmented. We have areas that are of the axon that are not covered by myelin sheath. These are called nodes of Ranvier. The gaps in the myelin sheath between adjacent Schwann cells is called nodes of Ranvier. So the nodes of Ranvier are gaps in the myelin sheath, and they are the sites where the axon collaterals can emerge. We have finished the glial cells of the peripheral nervous system. Now we are moving to talk about the glial cells of the central nervous system. They are 4 types: the oligodendrocytes, the astrocytes, the ependymal cells, and microglia. The oligodendrocytes, they produce myelin, and the oligodendrocytes processes, they wrap around the nerve fibers, and they insulate the nerve fibers. So they produce the myelin sheath, is similar to Schwann cells in peripheral nervous system. As we see in this diagram, these the oligodendrocytes, how they appear. This is cell body of the oligodendrocyte, and here we have the process of oligodendrocyte that traps around the nerve fiber and forming myelin sheath. It can wrap around many axons. The second type is the astrocytes, and the astrocytes, they are most abundant and highly branched glial cells. They maintain the blood-brain barrier, and they cling to neurons and their synaptic endings. and they can wrap around the capillaries, regulate their permeability. In this diagram, the astrocyte, how it looks like. It has many processes, highly branched glial cell. Its processes can wrap around the nerve fibers and also around the capillaries. The ependymal cells, they are epithelial-like cells. They range in shape from squamous to columnar, can be squamous or cuboidal or columnar. They are ranging in shape, and they line the ventricles of the brain and the central canal of the spinal cord. In this diagram, how it looks like the ependymal cells. This is the brain or spinal cord tissue, and on the other side we have the fluid-filled cavity filled with cerebrospinal fluid. And these ependymal cells, how it looks like, and it ranges from squamous to cuboidal in shape. In this diagram, it looks like more cuboidal. The microglia, the fourth type of glial cell in the central nervous system, they are small ovoid cells with spiny processes. They are monocyte-derived antigen-presenting cells of the central nervous system. They function as an immune defense for the central nervous system in the brain and spinal cord. They are monocyte-derived, unlike the other types of the glial cells, which are derived either from the neural tube or the neural crest, but the microglia is derived from the monocyte, circulating monocyte in the blood. This is how the microglial cell, how it appears. It's a small ovoid and it has many processes. They act as a antigen presenting cell of the central nervous system. Act as a phagocytic cell. They look for the invading microorganisms and the debris in the neural tissue of the central nervous system. The last topic in this lecture is the neuropil, and this is a fibrous intercellular network surrounding the cells of the central nervous system called the neuropil. Fibrous intercellular network surrounding the cells of the CNS called neuropil, and the neuropil consists of a network of cellular processes emerging from the neurons and the glial cells. In this image shows the neurons, the neuropil, and the common glial cells of the central nervous system. On the left side, the section is stained with hematoxylin and eosine stain. On the right side, we have a section stained with gold chloride and hematoxylin. Here in H&E stain, we can see N stands for nucleus and the cell body, perikaryon of the neuron, and also here we have nucleus and the body of the neuron, cell body of the neuron, perikaryon. MP stands for neuropil. This appears as here small fibers, and here G stands for glial cell. The nucleus of glial cell, actually it appears here, nuclei of the glial cells. On the right side in this section, which is stained with gold chloride and hematoxylin, also we'll see very clearly the neuropil, how it appears. It's better than in H&E stain. These fibers are the neuropil. These processes, okay, fibrous network. The neuropil is a fibrous network that consists of the cellular processes of the neurons and the glial cells. Both of them together they form this fibrous network called neuropil. MP stands for neuropil. N, this is the nucleus and cell body of neuron, and neuron. And G stands for glial cell. These dark ovoid structures, they are nuclei of the glial cells. So you notice the difference between this section and this section in distinguishing the neuropil. On the right side with gold chloride and hematoxylin, it's very clear that the neuropil is a fibrous processes or a fibrous network formed by the processes of the neurons, these are neurons, and the glial cells. So we have reached to the end of our lecture. In this lecture we have talked generally about the structure of the nerve tissue, about the neurons and the glial cells, and how the components or the parts of the neurons, axon, cell body, axon, and dendrites. And also we have talked about the types of the neurons, either anaxonic, bipolar, unipolar, or multipolar, and the types of synapses and the components of the synapse, presynaptic axon terminal, postsynaptic cell membrane, and synaptic cleft. Also have talked about the types of synapses, axo-axonic, axosomatic, axodendritic, and then we have talked about the glial cells in the peripheral nervous system, the Schwann cells, which forms myelin sheath, and identified or described the myelin sheath, which is lipoprotein, and we have it is a segmented. We have nodes of Ranvier, and also we talked about the satellite cells of the ganglia, which surround the perikaryon of the neurons in the ganglion, the spinal ganglia, and also we talked about the glia or neuroglia of the central nervous system, which is oligodendrocytes, the astrocytes, ependymal cells, and the microglia. And also we have identified the neuropil, which is a fibrous network in the central nervous system found between the neurons, and it's composed of the cellular processes of the neurons and the glial cells. By this we have finished our semester. This is the last lecture, and good luck for you in your examinations, and thank you very much.